So I guess one interesting thing is I started psychiatry. Psychiatry is ever so complex. I didn't realize, because when you think psychiatry, what do you all consider? What kind of diseases do you think about? Right, such as? Okay, that's one of them. Any others? Schizophrenia, anything else? Yeah, there's alcohol use disorders, there's bipolar disorder, and there's depression, right? Now, within each category, there are so many different subtypes of that disease. You can have depression with psychosis, where you actually hear and see people that are mentioning negative thoughts towards you. This is my first time seeing patients who are complaining of voices in the room, or patients who are hypomanic and think they're God, or patients that think they're, a trauma surgeon themselves. So I guess that just shows you how powerful the human mind is. If it compels itself to believe something, it will change its environment and reality to believe that, which is very interesting. The last thing I'll talk about very quickly is the topic of depression. Now, what does it mean? How do you diagnose depression? Because it's so common. And in fact, it's going to be the most common disease on this planet in the next 10 years. So any idea how we pick that up? Is it just sadness? Is that what we ask someone? Are you sad? Depression.
Is it how often is that, or like if they have panic attacks?
No, the thing is that's different. Anxiety and depression are correlated, but they're not causative for one another necessarily. Now, depression is usually, we have to put an arbitrary time frame because we need to diagnose things. So 2 weeks, or potentially sadness, but there's so much more to it. Any other signs and symptoms? What would you check up on yourself? If you're feeling low, because you might in the HSC year, it's a stressful year, there's a lot on your plate. What else would you want to check to see if it's just sadness or whether it's depression?
Maybe just perspective, like how you mindset. You can challenge your own perspective, and you can find yourself like, still stuck in the same mindset, it's not changing, you can't.
Yeah, okay, so it's quite refractory to change, right? Okay, interesting. Now, what if I told you didn't even need to be sad to be depressed? You don't. And we'll talk about that soon. Anything else?
Would you say that you have
That's actually a really important point. When we ask about depression, we don't just ask about sadness. One of the other main symptoms is a loss of interest in the stuff you know. So even if you have sadness or that other symptom I just mentioned, loss of interest, that is a hallmark of depression. The other symptoms are sleep and weight changes. So those are very common things you should always track with yourself to see if you're doing okay on this journey as well. Okay, cool. All right, let's jump into things. Mod 6 is very interesting because it's so much shorter than 5. You will breeze through it. I think about 5 weeks or so, you will probably be finished or getting towards the end of 6. Okay. Now, a quick talk about your exams. It's the 5th of March now. Raise your hand if you've done all the HSC Biology additional questions for Mod 5. Okay, that is fine. There's a lot of questions, but like I said, you are not seeing that exam until you show me that booklet finished. Okay, so if you come on the day of the exam and you don't finish that booklet, I'm sending you back home. Okay, so the date that I'm planning, the reason we finish that booklet by, is the week of the 19th to the 22nd. Okay, so you have about 2 weeks or so to do that. Is everyone happy with that? Okay, so this is your final message. Yes? Yeah, sure. You didn't do it good? Too fast? Okay, no worries. Most of you have an assessment. Do you have a biology assessment or not? Yeah. Is it on Mod 6? Is it on Mod 6?
Okay. Mod 6, second and third compartment.
Good enough. So you can study it as you go. Okay. So what you should have done is you shouldn't be cramming it for this exam. You should have prepared consistently throughout. So sitting this exam is just like, how are you going as a baseline progress? Don't stress about it too much. But if you do bad, then we do need to talk about it. Okay. Good. All right. Now, on to Mod 6, genetic change. This dot point or this entire module, has three main components. Does anyone know what those three main components are? Genetic change. You guys are saying the N word, that is the first component. Mutation, right? So the first entire section of this module is about mutation. And Brandon, I'll get you to give it a go. How would you define mutation?
I would say a mutation is a change in the structure of DNA, which can either be like point inversion, block mutation, or a rearrangement of the structure.
Very good. Very good, right? So a change in the DNA sequence, to sum it up, right? The two key things you need to know about mutation is what causes it, what are the effects. So that's how I would block it in my head. Mutation, what causes it, what are the effects. Now, the second part of this module is all about biotechnology. And look, Nick, give it a go and define biotechnology for me.
Technology that is used in biology. I'm not sure how I'll define it.
Look, that's a savvy answer, I like that. He's technically right, but you have to just fancy it out a little bit, right? Alcohol is a biotechnology because alcohol is produced using microscopic organisms, living organisms, which are yeast, right? They will ferment the glucose to produce the ethanol. So since we're using a living organism to produce something that is a biotechnology, the use of life or any of its byproducts in any human venture is a biotechnology. It could be in medicine, it could be in agriculture, it could be in industry. Does that make sense? Okay, so you'll be studying ancient, present, and future forms of biotechnology. And this is super broad. If you think about this, what are some other types of biotech that you can think of here? CRISPR-Cas9, it's insane. We are now getting to the point where we can cut and paste genes. And how are we doing it? We're looking at bacteria and how they fight their bugs. Bacteria can get infected with viruses. Okay, so that's actually how CRISPR was designed. We'll talk about that soon as well. Now, the last inquiry question is about genetic technology. What's the difference between biotech and genetic tech? How do you relate the two? Virginia, biotech versus genetic tech, what's the difference? Pranav?
Biotech is applied genetic technologies. Genetics, how we use it.
I want you guys to be thinking, right? When I say the word gene, doesn't that relate to life? So wouldn't biotech be the umbrella term and genetic tech be a subsection under biotech? That's how you need to see it. Does that make sense? So genetic technology is the use or modification of genes for any purpose. Okay? Now the last section is not just about genetic technology, but it's also about reproductive technology. Now Lukman, it's your time to shine. What are some reproductive technologies you know of?
Artificial insemination, artificial pollination, cloning.
Very good. Alyssa, what are some other cooler ones, right? Artificial pollination is picking pollen from the anther of one plant and putting it down the stigma style of another. What's a cooler form of reproductive tech?
IVF.
IVF is really interesting. We'll talk about that, right? We have literally surpassed genetic limitations of pregnancy. Normally, a female can't conceive after about 40 to 45. That's because they're born with all their ova, and by that age, they become quite damaged from radiation and mutagen exposure. Okay, mutagens being things that cause mutation. Now, we can freeze eggs, and we can use surrogates, right? You all saw that Kim Kardashian's pregnancy. I'm sure people know about surrogacy from that, right? So we've got to that point where we can control who gives birth, whose eggs we use, and when they can conceive, which is insane. Cloning is the one I was looking for. You can actually clone life. We can clone humans if we want to. We just don't do it yet. Okay, so our power right now as a species is getting to the point where we're starting to move outside of our genetic limitations. Does anyone know what the main issue with cloning is before I actually jump into the how you would start this module? Because you have to be interested in this to actually learn it and know it to a deep level.
Because you can't like clone
experiences.
Yeah, so it's basically a goal of learning. My goal is just to make an identical physical organism. What is a limitation? Think about Dolly the sheep. What happened to Dolly that kind of halted our progress in cloning? Yes.
I don't know what happened in Dolly, but the first question you asked is because like something might go wrong in the cloning that we might create, and
That is true. If you create new life with disease and mutation, that has huge ramifications, right? Anything else about Dolly specifically? Yeah, she died prematurely and she aged prematurely. And we'll talk about that very soon as to why that happened. Good. Now, as you can see here, content focus have underlined the key points. So I'm just going to ask people, and you're going to define it for me. So, Alyssa, I want you to define natural selection. It's not in this content focus, but I want you to define it regardless. What is your exam answer for natural selection? It's a quick. Lookman, give it a go. Guys who are online, if I ask you a question, answer it as soon as you can, okay?
I would say it's the process where populations adapt and change.
Okay, quite vague. Ramesh, I'm going to give you a go. Define natural selection, 1 mark.
I would say natural selection is a theory which states how animals and certain organisms adapt to their environment and through like different selection pressures adapt, and only a certain amount of the organism is left.
Okay, good. I like that, right? So, very good. So the key things on here when I ask you about evolution by natural selection is environmental selective pressures. Favorable organisms surviving, reproducing, and passing on these traits. Unfavorable organisms dying, and this altering allele frequency over time. That is what natural selection is. So I want to hear selective pressures, favorable, unfavorable, and change in genes over time, because that is what evolution is. Okay? Good. You guys need to get to high yield points. All right, we'll move on. All right, one word I do want to define here, because this will come up in your exams. What is biological diversity? We can shorten this to biodiversity. How would we define this? Because there are 9-mark questions in past exams that will ask you the effect of reproductive technologies on genetic species and ecosystem biodiversity. And students who don't know how to define this word stuff those questions up. So, room, biodiversity.
The range of alleles or genes.
Yeah, I would say, in fact, biodiversity is simply the variety of life on Earth. Summed up. Okay, good. Now, there are 3 levels to it. If you logically think about it, we can be talking about a variety of genes and alleles in a species. Does everyone agree? We can then branch out to look at the number of species in an ecosystem. And then we can branch out to look at the number of ecosystems on that planet. Do you see how there's 3 levels here? So the way I remember this is if I say the word species, if I say the word genetic biodiversity, I'm talking about the number of genes in a species. If I'm talking about species biodiversity, I'm talking about number of species. If I'm talking about ecosystem biodiversity, I'm talking about number of ecosystems. That is how you remember it. Does that make sense? Good. Very important term because it comes up a lot in your exams. Good. All right, let's start moving to our actual information. Now, you guys got up to naturally occurring mutagens, chemical mutagens. Which one did you get up to last lesson? What do your notes have? That's all right. I'm just going to quiz you and see, and I hope you've probably done the first stop notes. Okay. Now, when I say the word mutation, Ramit has defined it as a change in the dimension. It can be at the level of a base or the level of a chromosome, which is how we think about this. Now, I'm going to ask you how a mutation occurs. Because like I said, it's summer now, and although it's really wet and rainy, if you went to Bondi Beach in the sun for 1 hour, you would get about 20,000 point mutations in your DNA, right? So you can kind of infer mutation can occur through to an induced mechanism. It can be caused by something. And what do we call that term for any factor that causes mutation? Good, right? So that is a mutagen. But mutation happens another way as well. What is the other way? Think logic. Think module 5.
Yeah.
Exactly. There you go, right? So it can occur spontaneously, or it can be induced through a mutagen. Those are the ways in how a mutation can happen. Now, let me go with Kirthana. What phase in mitosis does DNA replicate? Because this is the most important phase. This is the phase where mutation occurs. What's our answer? Okay, guys who are online, if I ask you a question, answer it as soon as you can. Lookman, give it a go.
Interphase.
Good, right? And Ramit, would you know what phase in interphase?
Interphase 1?
There's not numbers to it, but I think you'll remember it's very good. There's a G0, S, and a G1 phase, or you can call it G1, S, and G2 phase, right? G2 stands for growth. Organelles will replicate, but the key thing is, in the S phase of interphase, S for synthesis, DNA is going to double. Okay? Good, and you can imagine it's 2 meters of DNA. We're going to make errors when we divide it. How do we fix those errors, by the way? What's the...
Splicing.
Not splicing. Splicing is, remember, with transcription. We splice to modify the RNA before it can go out. But, yeah, no worries.
Alyssa?
This is here online.
Ethan? Georgina? Martian?
Not here.
Jessica? Yeah. Dean?
Not here.
Hannah? Raya?
Raya's here online.
Ria? Tanisha? Tashia?
Oh, Tanisha's here, sorry.
Sorry, Tashia? Varun?
Yeah.
Isha? Abhishek?
Yeah.
Kushi? Here. Sachita? Lukman?
She's here.
Kirtana?
Keithan is also online.
Harshita? Gayatri? Martin?
Online.
And Raman.
He's here as well.
Guys, I'm just going to take photos in the class so quickly.
Thanks, Keithan. All right, cool. So let's talk about your point mutations. Let's talk about causal mutation now. Now, when the syllabus breaks down the different kinds of mutation, there are 3 different types you need to know. So there's physical, chemical, and naturally occurring. Those are your 3 main classes of mutagens. We'll start with physical mutagens. What are some examples that we know of? Okay, what does this word ionizing mean to us? Bro? Any ideas? No? Did you not learn this last week? Not quite. What does ionizing mean, everyone?
In terms of chemistry, like something associated.
This is the thing I will have to understand. Physics, chemistry, biology are different sides of the same coin. If you learn something in chemistry, it carries on to biology as well. So, any ideas?
I don't know the exact name of it, but it's pretty.
Okay, we'll talk about this, okay? So, when we mention radiation, the specific radiation we're talking about is electromagnetic radiation. The reason we call it electromagnetic is because if you zoom into the wave, it actually consists of an electric and magnetic field that are continuously propagating one another. Okay? That's the physics of things. That is why it's called an electromagnetic wave. Now, these waves are all invisible except for this tiny little patch in this entire spectrum, which is visible light. You have to think about this. The human body has designed itself to pick up very specific wavelengths meaning size of the wave, right, within this huge spectrum. We only pick this up, guys. We are actually blind to 98% of what's going on in the world around us, right? We only see the visible light part. Everything outside is still radiation, okay? And as you can see, we have different kinds of waves, and I'm sure you've heard of gamma rays. Anyone watch the Hulk? Right? You learn through the world around, right? So, as you know, it's bad for you. Whereas radio waves are being sent through telephone towers. Radio waves are passing through us right now, right? So those are very low energy. Now what happens is when you have a very high energy wave, it can have enough energy that it collides with atoms and it will knock an electron off. That is called ionization. Because when you knock an electron off, we create a charged atom or an ion. Does that make sense? So high energy waves are known as ionizing waves. Now, I'm not here to teach you physics, but you do need to know this tiny formula so everything makes sense. Okay? Don't worry about H. H is a constant, but this formula is telling you the energy of a wave is directly proportional to F. What would F be for a wave? You all did basic science. Frequency, right? So the higher the frequency, the higher the energy of the wave. Now, specifically, the ionizing frequencies are UV, X-rays, and gamma. Those are the three you must know. All the other wavelengths are non-ionizing. They don't have enough energy to knock off electrons, and thus they won't damage atoms. Okay? Now, question. 60% of your body is made up of a particular molecule. Good. 60% of your body weight is water. So if I was to get a gamma gun and shoot an arm, what's the most common molecule that I would hit? Water, right? Very good. Now, what did I say? If I use an ionizing wavelength of radiation, what will that do to the water molecules? Take an electron, right? Now, you all learned in chemistry, hopefully, everything in the universe, every atom, wants a stable octet configuration. It wants 8 electrons in its outer shell. Chemistry is literally a mini game of every single element aiming to get to 8 electrons, right? Now, if I was to take an electron of the stable water molecule, what would it want to do? Try find another. Now, what it can do is it will go and react with other molecules, ripping their electrons off. And what if it goes to your DNA and it rips an electron off DNA? What do you think that could do to the structure of DNA? It could change DNA. And what do we call it when we change DNA? There you go. So that is how radiation causes mutation. We'll write down the exam answer, but that is the main way. There is a simpler way though, everyone. If I was very lucky and I shot him with that ionizing gun, I could directly damage DNA, right? That X-ray or gamma ray can directly just knock an electron off DNA. But in real life, the most likely thing that happens is we form water molecules. Here's your little diagram. This is exactly what I'm talking about. So I'm talking about the indirect effect, which is most important, right? We will react with a water molecule. As you can see, this is water. You knocked off an electron, and the water is now going and damaging DNA, and thus causing mutation. And the less common method is we directly damage molecules in DNA through radiation. Question?
So the electron is not there. Where does the electron go?
This is a thing, right? All around you, there are electrons that are moving around. There's waves, there's fields of all particles. So it goes into a bit of physics here, which is that electrons are both a wave and a particle. And if you ask me where are the light rays in this room, would I be able to tell you where this light ray is going? No, because in the same way, it's hard to point at where an electron is at a point in time. It's a bit mind-blowing, but we now know that electrons around an atom don't exist as moving balls. That's what you learned in junior school. They have a cloud-like distribution where we can say there's a high probability an electron will be there and a low probability. Yes? Yeah, so now we're going to add terms to the things we just spoke about. So when I said that we form highly reactive atoms that want to get those 8 electrons back, those are free radicals, right? What do radicals do? They're extremists. Free radicals are chemical extremists. They will do anything it takes to get the 8 electrons again. Does that make sense? So to sum things up, water, when we damage water molecules, it's going to form free radicals. So I'll just label this as free radicals. And then free radicals will then go and react with DNA, causing mutation. Good. Any questions so far about your physical mutagen? That is your main physical mutagen. Ionizing electromagnetic radiation. Okay? There are 3 you need to know, and now you know exactly how to damage and change DNA. Any questions so far? Good. All right. Now, very quick talk. We spoke about electromagnetic radiation, but there's also particle radiation. You guys heard of alpha and beta particles before, right? Now, we're not going to go through this in detail because the biology syllabus doesn't require you to know this, but just know there are particle forms of radiation too. Now, I want you to imagine if you were shot with a wave, you were shot with a particle which has mass. A wave has no mass. Which one is more likely to knock an electron off? Think about this. If you're going to be tackled by a ghost which has no mass, or you were to be tackled by an NRL player, who would knock you out faster? Yeah. So in this case, particle radiation, which has mass, will knock electrons off at a much greater rate. It has a much greater ionizing capacity. So what we're going to write here is particulate radiation has mass, so it has a high ionizing ability. Whereas if you're a ghost, you can pass through walls easier, right? So waves have a higher penetrance. As you can see, this gamma particle, which is a gamma wave, is able to pass through surfaces. Good. That's just a small point to understand. But again, this is much more physics, so we're going to move on. All right. Now, one specific point that I will mention, this is only for trials, so maybe asterisk this in your notes and write trials, is a specific way that UV radiation damages DNA. Now, Lookman, what's a purine and what's a pyrimidine?
Purines are double-ringed bases and include adenine and guanine, and pyrimidines are single-ringed, including cytosine and thymine.
Amazing. Good job. All right. The way I remember this, it's weird. I remember pure, and I remember agriculture. I don't know why, right? Like close to agriculture. So AG for agriculture, so purines are your double-ringed A and G bases. Find whatever works for you, just have a weird mnemonics, okay? Good. So what can happen is with pyrimidines, now we understand pyrimidines are your C's and your T's. What UV radiation does is, this is a non-ionizing effect, by the way. This is independent to UV radiation knocking electrons off things. UV radiation specifically can form unwanted bonds between 2 pyrimidines. You see how these are 2 pyrimidines on the same side of DNA. We're not meant to form direct bonds between bases. It's meant to be base bonded to a sugar, bonded to a, then bonded back to a sugar, and then to a base. But we formed a direct bond between 2 pyrimidines, right? This is known as pyrimidine dimerization. Okay? Dimer meaning 2. Di is 2, right? So that is a unique function of UV radiation. Okay, good. Any questions so far? Good. All right. Let's talk about chemical mutagens now. Now, chemical mutagens are broad. I'm just going to ask people for some examples. Don't look at this because there's way more cooler examples. I'm just going to go across online students Make sure you answer me when I ask you. So, Martin, what's an example of a chemical mutagen you know of? Any examples? Think about any chemicals that cause cancer. What would you not want to ingest or be in contact with? Lead, heavy metals, right? Very logical. I'm not asking you guys fancy high-level physics, chemistry, just what causes cancer? Lead, very good. So heavy metals can. Exactly. Any other examples you know of? Phosphorus doesn't. I get where you're thinking. It looks quite funky, the chemical, but we use it in fertilizer, so we need it for life. Any other chemicals? Who likes charcoal chicken? I had it like 2 days ago. Who's had it recently? You ingested carcinogens. You ingested cancer-causing chemicals. The reason being, you see that black soot, that carbon solid on charcoal chicken? When it goes to your large intestine, it's like sandpaper. It rubs on the colon, which is your large intestine. What does it do to cells? They die. And then what has to happen to replace those cells? And what happens to the S phase and interphase of mitosis? And so what can happen if that doesn't go right? Which can lead to, there you go. So you get the link now, charcoal chicken cancer, right? Doesn't mean stop eating it, everything in moderation, right? Who likes sushi? I had sushi yesterday. Sushi, smoked food, has a high risk of gastric cancer. This is rare. Japanese people are one of the longest living populations on the planet. They have the most octogenarians, which I think is 80-plus-year-old people, right? But if they do get cancer, they get gastric cancer because of the smoked food that they ingest. Okay, good. Any other cancer-causing substances? Aramis, any ones that you know of?
Did you mean chemical ones or?
Yeah, specifically chemical ones.
I was gonna say carcinogens as well, but even like cigarettes.
Cigarettes, very good. I was hoping someone would say cigarettes because we're talking about charcoal chicken, but cigarettes is the number one thing on this planet, number one chemical that can cause cancer. Now, I've said a word which I've kind of brushed, which is this versus this. What's the difference? Okay, carcinogen causes cancer, and a mutagen causes. Okay, so how are they related? Does that mean all mutagens are carcinogens? Does that mean all carcinogens are mutagens? Lookman?
Well, carcinogens is caused when a mutation accelerates the cell cycle, and that causes uncontrollable cell division.
Perfect. Okay, did you guys get that? A mutagen is any factor that will cause a mutation, right? Now you have to think about this. A mutation in your eye color gene will change your eye color. Mutation in a height gene will change your height. But if you mutate a cell cycle gene, you can cause increased rate of division, which is a hallmark of cancer. So what that means is a mutagen, when it causes a mutation, only if that mutagen affects a gene associated with cell division can it cause cancer. So what that means is technically all mutagens are carcinogens. They can be. It depends what gene they hit. Does that make sense? So keeping our right is if a mutagen mutates a cell cycle gene, that's going to lead to increased cell division, and that is the hallmark of cancer. You guys are going to learn something very important this module, which is why we still can't cure cancer, right? Because there's a lot of people that believe a lot of alternative therapies, curative therapies exist for cancer. And you're going to understand why it is so hard to solve this thing. Because cancer is not just one disease. And we'll talk about that very soon. Okay? Good. Does that make sense to everyone? Okay? Only a mutation in a cell cycle gene will increase risk of cancer. Mutation in any other gene will just change the phenotype of that gene. Okay? Very good. All right. Now, a few ones I want to talk about that you need to know are listed here. Now, with cigarettes, there are hundreds of carcinogens. There's heavy metals, there's so many different types of organic compounds that are carcinogenic. But the main one you should know is polycyclic hydrocarbons. What this means is structures of carbon and hydrogen that exist in a ring-like shape. Okay? So those are carcinogenic. That's the main carcinogen in cigarette smoke. Okay? Good. We've spoken about smoked meats. Meat is high in nitrogen. It's high in amines. If you do chemistry, you know amines are the bases, and they're consist of hydrogen and nitrogen as well. So you can actually combine the two and are very high temperatures, and that's also carcinogenic as well. In the same way I described, it's going to irritate the intestines and increase risk of cancer as well. Now, asbestos is a very interesting one because it's not too much of an issue now, but it was an issue back in the day, in the 1940s. We used to use asbestos in housing insulation. And what happened is 60 years later, people started getting this fatal type of cancer, which has a less than 7% survival rate, called mesothelioma. If you ever see the word oma on a medical report, that's bad news. It's a tumor, right? Mesothelioma, the mesothelium is the lining of the lungs. So mesothelioma is a cancer arising from the lining of the lungs. Now, why did it take 60 years from asbestos exposure to get this disease? Why don't you just get cancer a year later? Any ideas? It's because cancer is not just one mutation. You don't just mutate one gene and the cell continues to divide forever. You need to mutate many consecutive genes associated with cell division. And as you can imagine, that takes time, right? And so it takes on average, usually about 30 to 60 years from exposure to a carcinogen to getting the cancer. You need to constantly be exposed to the carcinogen, okay? That is also why smoking, you don't get cancer the next day after you smoke. It takes years. You need to keep affecting and mutating cell cycle genes. And that's kind of the issue now with vaping, which is we don't know. We literally have no data. It's been around for like 10, 15 years. We don't know if in 60 years we'll get about some increase of a rare cancer of the lining of the lungs. We just don't know, right? So that's a really important point to note, that from carcinogen exposure, it takes time to get cancer. Okay? Good. Now, the key point here is, how do chemical mutagens work? Because we said physical mutagens knock electrons off. How do chemical mutagens cause cancer? There's one overarching way. What? Yeah, they affect replication, but more specifically, yeah? Yes, that's one way, but the most common way is all these chemicals, they appear structurally similar to DNA. Appear structurally similar. Now, when you appear structurally similar to DNA, during DNA replication, when DNA polymerase is picking up free-floating nucleotides, what can it accidentally pick up instead? One of the mutagens. Whether it be a heavy metal, whether it be a polycyclic hydrocarbon. And what could it accidentally do? Insert it into DNA. And what does that do to the DNA sequence? Mutation. So that is the main way chemical mutagens work. Physical mutagens knock off electrons, chemical mutagens incorporate themselves into DNA. These are overarching points. There are obviously exceptions to the rule, but that's the principle you need to understand. Yes?
How is chemical mutagens present in the nucleus?
How are they present in the nucleus? It's very complicated, because then when you start talking about, well, how do cells move things in and out? It's through channel proteins. and channel proteins can accidentally bring these chemicals from the extracellular space in. So it just occurs, it's very complicated if you start talking about channels and everything. So, yeah, good. Any questions at all? Ramith, Lukman, Melissa? No? Right, good. We'll move on. We'll talk about biological mutagens. Now, what's an example of a biological? I find this the most interesting. It's super cool. I'll give you a case so you understand how it can affect you. It's actually common for kids your age to get sick, just runny nose, sore throat, bit of a tummy upset, and swollen lymph nodes. And then within 2 years, a small percentage of them will get lymphoma, which is cancer of the lymph nodes. This is young people I'm talking about, your age, my age. Now, what caused that to happen? You look back, how did that young person get cancer? Because getting cancer at 20 is not normal. Getting cancer at 80 is normal because you've been exposed to so much mutagen in your life. Cancer at 20 is not normal. So if you're the doctor, and that's the history we've got, what disease would you be worried about? Very hard question, so I don't expect you to know, but any guesses as to what kind of bug did that to the person? There is one class of bug that can cause cancer. What bugs do you all know? Shout it out. There's bacteria, but it's another bugs that cause disease. You're in a pandemic caused by something other than a bacteria. What is it? Yes, so viruses, right? Viruses can insert themselves into your DNA and change your DNA sequence. So what I just described was a virus called the Epstein-Barr virus, right? And I can guarantee all of you, by the time you turn 20 or 21, 90% of you will be infected with this virus, okay? And majority of the time you just get a mild fever, right? Mild fever, swollen lymph nodes. You might have already had it, right? But a small percentage of the population, that virus remains and it damages the DNA sequence by integrating its own DNA in. And that causes potentially cancer of the lymph nodes. Okay? So that's one way in which a biological mutagen causes cancer. So one of the key examples is viruses. Does that make sense? So highlight it, underline it, viruses. There is one other virus. This is one of my favorite diseases. When I was doing HSC Biology, this was one of the reasons I wanted to do medicine. There is this really cool virus. It's the only virus that can do this. Usually viruses evade the immune system, right? COVID tries to escape immunity. This is the one virus that is so badass, it will attack your immune system head on. It will win every single time. And the only way to cure it, well, you can't cure it, the only way to prevent its progression is using medication therapy. What virus am I talking about? HIV. HIV infects immune cells. Its goal is it's literally like a robber that's trying to rob a police station. That's what HIV is doing. Okay? Now, in its venture, what happens is HIV, this is why it's incurable, it has an enzyme called integrase, which allows it to integrate its DNA into host cells. That causes mutation. Okay? And that's why you can never cure HIV. Because what if the human body, so imagine you destroy all the HIV particles in the human body. You still have the HIV DNA or RNA in your genes. So when your transcription and translation accidentally makes protein out of that gene, what will it produce? It will make HIV, your own cells. And that's why it can never go away, right? I'm going into a lot of detail here, but I just want you to get some pictures and examples. So now hopefully you never forget, viruses are one of the main examples of biological mutagen. Yes?
So do plasmids not recognize?
Well, I wish they did. They don't, unfortunately. Yeah, they don't. Yeah. Good. All right. Now, what percentage of your DNA is actually coding for protein?
Isn't it 90, 95? Close to 95 is non-coding. And others coding?
Lookman's got his hand up. Lookman?
Coding is around 2%.
Yeah.
Yeah, so coding DNA, very good, is only 2% of your entire genome. That means 98% is non-coding. And one of our theories as to why we have so much non-coding DNA is our ancestors were infected with viruses like HIV, which integrated their gene sequences into the DNA. And what happens when you have a child? You pass on your genes, including that stored viral DNA. So in fact, that 98% of your DNA, majority of it, is ancient viral DNA from your ancestors being infected, which is pretty cool. But that explains why only 2% actually makes things. Okay, good. All right, we're gonna talk about some other biological mutagens. Now, one of the coolest things I learned in one of my first lectures in medical school was one of the ways to live longer is to eat less. And numerous studies showed this to the point where being on a slight caloric restriction added up to 10 years of life. That's insane. Imagine getting a free card to add 10 years to your life. It's a free boost, right? The way this happens is when you eat things, your metabolic rate increases. Do you guys all agree? What is metabolism? It's the breakdown and the build-up. It's the chemical reactions in the body. When you eat something, your metabolism or the chemical reactions in your body increase. Now, when chemical reactions increase, what happens to the amount of waste that's produced? Increases. And some of that waste is free radical species. What are free radicals? We just spoke about this. Kashia, free radicals, define it. Good. Highly reactive species that has an unpaired electron, right? So it has one electron that isn't paired to another one. Okay? So one of the waste products you produce are free radicals. When you produce free radicals, what do they like to do? They like to react and become stable again. So what can they do to DNA? Exactly, right? So you guys getting the pattern here? If you eat more, metabolism, if you increase consumption, you're going to increase metabolism. And what the syllabus wants you to state is you'll increase end products of metabolism, which include free radicals. Free radicals. And that's going to cause DNA damage. Now, it's not just with you. I want you all to realize this. End products of metabolism is what the syllabus has specifically listed. If you don't use this word, you won't get any marks, okay? But to understand this, it's not just human metabolism. Other organisms like fungi produce end products of metabolism that are carcinogenic to us as well. So it's not just our end products of metabolism. There's other organisms like fungi and bacteria which produce end products of metabolism that are also carcinogenic or mutagenic to us. Okay, so just remember, it's not just human end products, it's all types of organisms. The end products of metabolism are harmful to human DNA. Good. Does that make sense so far? Good. I thought that was really cool. And I kind of have incorporated that now, where I don't want to bulk all the time and try to look like a sumo wrestler because you'll die from it. But eating a little bit less or eating until you're satisfied makes you live longer, which is an interesting fact. All right. Any questions so far? What does this word mean? Brushed over this. Transposon. Anyone read about it? Yeah, very good. They are DNA sequences. Tell me more. Pardon? Okay, so we'll talk about it now. So I want you to imagine that this is your DNA. And what percentage did we say I was actually coding and actually made protein? Okay, so we'll say this 2% actually makes stuff. Everything else is not coding. Okay? Now, transposons are sequences of DNA that jump. And the two words I'd like you to remember is they're jumping genes. Jumping genes. Now, if a DNA sequence jumps from point A to point B, is that going to affect any proteins at all? No. This transposon is just jumping around happily. But if it happens to jump into the 2% that actually makes protein, will it change the DNA sequence of that particular gene? Will that change the protein being made? That's a mutation. So transposons are DNA sequences in your genome right now that are just jumping along a chromosome. And if they happen to jump from non-coding to coding DNA, they'll affect the transcription translation of that gene. That's a mutation. So keyword here, transposon jumping gene, full stop. Any questions so far? Good. Good. You're all doing well. All right. Happy to move on? Now you might notice I'm going a little bit fast, but that's because this is a lower still part of the module. They're not going to ask you many questions about all the different mutagens, and those are things that you just read and you know. I want to get to the part of the module which is application heavy, which is what your exam is based on. Okay? Good. I'll just do one question. Checkpoints. Here's do question 3 for me. I've got it up. Hopefully you can all read it. This is the kind of exam questions you'll get. The HSC will never ask you, sorry, we know the screen is. Okay. The HSC won't ask you questions that are directly content related. It's going to be application questions. Every single one you do. Have a read. Online students, when you get an answer, hit the raise hand icons, and I'll ask one of you as well. So hold on to a pen and you get your answer, put the pen down so I know you've got an answer. Surely someone's got an answer by now. Is it a loaded question? You have 30 more seconds. Alyssa, have you got an answer?
No.
Good. All right. Kirtana, you got an answer?
No.
All right, good. Raya? Any answers yet?
No.
All right. All right, let's start. Hands down. Who got A as an answer? Who got D as an answer? Who got C? C? Who got B as an answer? What did you get, Ramin? What was your answer?
I got C.
Okay. So C is... Okay. No one got the answer, which is good, which means you're all learning. All right. Let's do this question. You see this complicated flowchart, but it's actually quite simple. It's genes making proteins or polypeptides, which in this case are enzymes, which are converting one chemical to another chemical. This is what happens in the human body, right? This is happening, this flowchart, but imagine scaling it out billionfold. That is the human body, right? So just understand how complex we're talking. This is the simplest depiction of what's going on. Now, they're asking which of the following is most correct. Now, just be cautious, because the question might ask which is not true. If you don't frame the question right, you will not get the answer correct. So be very attentive to what it's asking you. All right, so which is correct? Ornithine must be an amino acid because it was produced from gene A. Now, I'll tell you this, ornithine is an amino acid, but is the justification right? Is ornithine produced from gene A? It's not directly made from gene A. Gene A is making an enzyme which is converting one reagent or reactant into a product, right? It doesn't have to be a protein. So I'd say A is not correct. Makes sense? The fungus with a dysfunctional mutation of gene B. So let's just get a red or a pen and knock out gene B. Would be able to grow in a cell culture with ornithine. Now, if you knock out gene B, everyone, what's the flow-on effect? What happens to enzyme B? Good. So if you've been given ornithine, can you convert that to citrulline? No. So this fungus cannot go about its normal reactions for life. The answer is not B. What about C, which is what most of us seem to have picked? A fungus with a dysfunctional mutation in gene C could produce arginine. Remember, arginine is an end product. This is arginine, right? When supplemented with both ornithine and citrulline. If you've knocked out gene C, what can't you make? Can't make enzyme C. So even if you have ornithine and citrulline, can you convert that to arginine? Across this step out. So that leads us to what are the chemical processes that are producing enzyme A, B, and C? Transcription and translation. I was just asking you, how is an enzyme made, but in a really fancy way, right? Now, hopefully this question taught you something. It's that sometimes in the HSC exams, the correct answer is the vaguest answer, okay? And I've often seen that the case with questions 16 to 20, where there's some small minor issue every other option. Remember, the goal here is not to pick necessarily the correct answer, but it's the best answer. And do we agree in this context, the best answer is D? It's super vague, which makes it a crap answer, but it is correct. Okay, good. All right, we'll move on. We'll do the rest of the questions for homework. These are all really cool questions. In fact, there is a bug, and this should be under biological mutagens. I didn't mention this earlier, but it's a really good example because it's an Australian example. There was a doctor who wanted to prove that a bacteria called H. pylori causes stomach ulcers and stomach cancer. Do we know how he proved the association with H. pylori and stomach ulcers? So back in the day, we used to think it was just due to functional changes or changes in acid content. Do we know what he did to prove? Yeah, he literally did a self-experiment. He took the bug, literally just down the glass with the bug, and he got stomach ulcers a couple of weeks later. And it was arguably the stupidest thing someone could do, but he got a Nobel Prize for stomach ulcers. It's worth it, right? So now his name is immortalized in medicine. So that's one of the most famous Australian medical stories, but it was an Aussie doctor who did this. Good. All right, let's move on to now the processes and effects of mutation. We finished cause. That's it. Okay, I'm not going to go back to causes of mutations. If you have any questions, feel free to ask it now or later. But we will jump to processes and effects. And since we're at the 12:05 mark, I will give you all your 5-minute break now as well. So we'll recap at 12:10. If you have any questions, you can also ask me this time. Otherwise, stand up, stretch, take a 5-minute break, and recap at 12:10. Random question?
Hi Prithvi, I was just wondering if when can I get the module 6 booklet? Because I think I got the wrong one. I got 5 instead.
Oh, wait, you got access to 5?
Yeah. Oh well, like the
Online access?
yeah.
Okay. You should get printed access. So usually what happens, Ramith, is you get a call from admin after your first class, and then you can request to get the mod 5 and 6 booklets printed and sent to you. And that's what I would prefer, because then you can note take on the booklets and everything. Does that make sense?
Right, that sounds good.
You'll also get online access on the edX website once you're a student, because you get login details.
Yeah.
And then you would be able to access the booklets as well.
I'm looking forward to enrolling.
Okay, cool. Yeah, so I guess after the class, we can sort that out. So just stay back after class. Just hit the raise hand icon. We'll talk about that.
Sounds
good. Hey. Good.
So I just wanted to ask you if I should be doing other cancer tests as well.
How are you preparing? Because that's very
I'm
deep.
using a program called Medify.
Medify is good. Good. All right. And how are your marks here for this going?
I haven't gone to much, but I think the social reasoning is my weakest second.
I would say watch a ton of YouTube videos. There's so much free content on how to do well in the UCAT exam. The UCAT is the same as the UK CAT, which has been in the UK for ages. So UK CAT, how to solve questions, and they'll take you exactly how to do it. Do a ton of Medify questions. Treat it like 2 minutes of a HSC subject. And then what you need to do is you find out areas where you're weak, where even free videos and all this is not helping. Then I would say get one-on-one help from someone who can help you with that weak section. So you might only need 1 or 2 classes or things like that. That would be my game plan. Don't join those big, 100-person courses, which are like, I know Medits is like $800 per day. And I remember doing it. I didn't learn anything because it's so generalized and you sit test at the end. The UCAT is a very personal test where you need to do well in all the sections. That would be my game plan. If you're starting out, it's a bit late though. It's not sure. Good. Okay. So usually students would start in Jan. And just know ATAR is also very important. You need to be aiming for over 95 for Western, over 99.6 for you must study. Right? Which school do you want to go? Blackfields. You have to be an outlier, you know that, right? No one gets in from Macquarie Fields usually, so you're going to have to be way ahead of your group, which means you have to be very competitive. It's tough. So, yeah. So keep me updated on your ranks. Do you know your ranks at the moment? Find out, because it's also very important. Okay? Alright.
Oh, wait, I've got a question. So, you know the test, how it's on the 19th?
Yeah.
The day before, we have like our bio exam, and then the next week, I have 3 exams, and they also have like 2 exams, and then next week they have like...
Let's see what day week is. If you'd like to consider it a week early, would you like that? So, you're saying 19th, you've got exams on Monday, like 21st, right?
Yeah, for next week.
Yeah, okay. What you guys can do then is you can sit the exam on the 12th, okay? And you can watch the recording for the class. So you can come to class, but you can go to another section of the exam, And then on the 19th, you can get like a study break for your exams. Does that work out for you?
Yeah.
Yeah?
Having this thing next week, I'm assuming you go through a second question, mutations. That's the
Well, up to
The only issue is you guys are doing chemistry as well, so I can't reduce the exam after because you're with some chemistry learning. You could come in at night and sit the exam. I would honestly say, because if I was you, what I would do is I'd sit the exam the week before and just watch the recording for that lesson so you don't miss out. I mean, you can ask me questions via Teams if you need to.
Yeah. Because, I want to do my exam during.
It's either that or you guys sit the exam. Yeah, I would personally think if you have an exam block, you should probably focus on the exam because that affects your actual rank and everything, right? Alright, how about we do sit the exam 1.5 hours. What we can do is if it's a 1.5 hour exam, the first 30 minutes I can go through the stuff that's in your exam for your course and stuff. We'll focus on that. And then the 1.5 hours you see your exam, I'll go through some other content, etc. Yeah. What is your exam about? Have you guys sent me? Good. Give me a rundown. What's it based on?
We had university equipment,
They did this last year.
Yeah.
Yeah. Did you guys get last year's like work resources and stuff? Okay, I can find it. But you guys have to let me know. So send me a message asking for like past students' work. They said, from what I recall, the exam not really test actual biology stuff very much. You're looking at plasmid DNA, is that your iodine and stuff? So they got some weird questions as well. Did you have access to last year's exam?
No.
Yeah, I can give you that, but don't share it outside of you agree, okay? Because that's not fair for other hosts and students. But how does it go? How do you see how the exam is?
Apparently there's like a video that should be ongoing in the exam. We have to.
Exactly. This electrophoresis question, but the electrophoresis plasmid DNA of different sizes. So I'll show you. I'll give you a work. Yeah. All right, cool. What do you want me to go through the next lesson?
I'll just get this as a mutation now.
We'll go through that after class. It's 12 now. Everyone, just ask them to come in. Robert, did you have a question? All right, let's jump back in. All right. How are you guys all going with your HSC study? How's the progress? Anyone feeling a bit burnt out? No, you're all chugging along well, doing all right? Okay. What do you do on the days you don't feel like doing work? Reasonable. I think that's actually not too bad because when I don't feel like doing work, I would do it because it's good because if you force yourself on those days, you're more likely to burn out later. So if you find out that you're overwhelmed, it's good to take a couple of days off. But what else do you do to keep yourself accountable? Let's say there's too many days you're not doing work. What do you do then? You work? If you just rely on your willpower to work when you want to, and then when you don't work, you're not going to work much. And your baseline, you kind of realize from your marks, will not be where you want to be. So how do you push yourself to work more consistently? Discipline's hard. What do? Just like Batman? Just discipline? Pardon? Schedule, yeah? Good. Very good. That's one really good way to do it. So, anything else? Yeah, so I'll tell you guys what I'm doing, right? since this is my final year, I've got to pass, I've got to smash my exams, right? And I don't want to just pass. I have like an internal drive to do well. So what I did was I found the smartest person in my cohort. She came first in the state for mathematics. She got 9.95 ATAR as well. Really smart. And I basically formed an alliance where we study together. And we do, like, we have 500 cases, we do 20 cases every study session. That way, I have to rock up to the study session knowing the cases, otherwise I've let the other person down. So externalize accountability is a key point, and make sure that there is a cost of not doing your work, that there is some form of punishment that is not. You're basically conditioning yourself. If you do that, you will get work done, right? So make sure you have systems in place, that's what I mean. Don't rely on willpower, motivation, all that's BS. Okay, your serious marathon is about consistency more than how driven or how high of an ATAR you have. Okay? Good. Who wants to do medicine or law again? This is particularly important for those of you because the ATAR is insanely high. It's out of proportion high to do with even the difficulty of the course. It's just really hard to get in. And with courses like law, Wollongong law is very different to UC law. I'll tell you that. And the ATAR cutoff reflects that, right? So don't aim for any law school, etc. Okay? Good. If you're doing other courses, just get the ATAR cutoff you need. I think 90 should be decent to get into the engineering and stuff. Aim for that, and yeah, you'll be a bit more smooth sailing than those who are sitting medicine and law exams. Good. All right, let's come back to this. So Ramit mentioned a really important point, which is when we're talking about mutation, there's two types: mutation, point, and chromosomal. Now, the word point just means a point in the DNA sequence. Remember, the smallest unit of DNA is the level of a base. So when we say point mutation, we're talking about a base mutation. Make sense? Good. We're going to talk solely about base mutations for a while. So I want us to zoom in and follow and look at base mutations. Now, what are the things you could do to a base? If you're looking at a base in a DNA sequence, what are the different ways you could change that sequence at the level of one base? You could insert one base, delete one base, change a base. There you go. You guys have identified the 3 ways you can cause a point mutation, right? Now, I want us to really understand this point because when we talk about base mutation, there are base substitution mutations. And then we have a separate category, base addition and deletion. And yeah, this word called frameshift, which a few of you mentioned, but students often struggle to explain to me why it's called a frameshift. Who wants to give it a go?
It shifts the like the frame of the, and then like the reading sequence is altered when you add or delete the base. Especially when you're like reading the code.
Good, that's the word I was looking for, right? So I want you to use a little thought experiment to understand this, right? As you know, the tRNA sequence, the mRNA sequence works by codons, right? For every mRNA codon, there'll be a tRNA anticodon, which will attach an amino acid. Which means for every mRNA codon, we code for one amino acid. Is everyone with me? So I'm going to write mRNA codons. I'm going to write C, CU, AAA, TTT, and we'll just write, sorry, I should write T, it's mRNA. UUU, and I'll write GGG here. Okay? Now I want you to imagine that we go ahead and we add AA. What does that do to the first codon? The first codon now changes from CCU to CC. The second codon changes from AAA to AAA because that UAA, apologies, UAA. That U then moves on to the next position, so it's UAA. And this next codon sequence will now change to AUU. And the next will change to? You see how the addition of one base has changed the entire sequential codon sequence thereafter. Key point, thereafter. Sequences before that addition are unchanged, right? It's only all the codon sequences after that mutation which are all frame shifted. The reason we say the word frame shift is it changes the reading frame of DNA. You know how you read in words? You don't say W-O-R-D word, you just say word because you understand it in accumulation. In the same way, the mRNA sequence, it reads it in the codon sequence, is read as codons, which are words. So what's going to happen now is the entire reading frame has changed, right? So we call it a frame shift mutation. Okay, good. Does that make sense to everyone? The key way you explain this is you say this alters the reading frame of DNA thereafter the mutation. Every codon sequence thereafter changes, and thus the amino acids thereafter will change. Good. The exam answers are all here, okay? So read that in your own time. But that's just a quick intro. We'll come back to frame shift. Okay? Good. Very good. Any questions so far? All right, good. So frame shift consists of addition or deletion, and base substitution mutations are the only category. Does everyone see that? Now, the other thing I want you to understand is a base substitution mutation has many different subtypes in itself. 3 main subtypes. If I substitute one base with another, so I want you to imagine I've got an AAA, and I substitute that with AGA. Okay? Does everyone agree I haven't affected the reading frame, right? I haven't pushed any base to the next codon. That hasn't happened. But what can happen here? What are the potential effects that can arise from this? Okay, so number 1, you can make a different amino acid. Triangle for change, changed amino acid. What else can happen? Good. Why could it still make the same amino acid? It's a different codon. Good. There's 64 different codons that code for 20 amino acids. There's an overlap, where that means many different codons will code for the same amino acid. Like English, many different words have the same meaning, right? Very similar principle there. Why is that the case? Does anyone understand why? Why did biology and evolution work that way? That we have 64 different words code for 20 different meanings or amino acids. Why not just make it 2020? Life's easy that way. Anyone? I'm going to come back to this question because I'm not spoon feeding you the answer. Think critically, and in 10 minutes, I want someone to have thought of the answer. Okay? Good. What's the final thing that can happen? Sorry, so we said the same amino acid. I should write same. Changed amino acid, and good. You can be a stop codon. Very good. What does a stop codon code for? You know how we said codon codes for an amino acid? What does the stop codon code for? Good, but actually codes for a substance or a particle that will bind to cut. You're thinking about the start codon, yeah, which codes for methionine. It codes for a protein called release factor. Has anyone seen that in the textbooks? This is extension knowledge, right? But it codes for a specific kind of protein that's going to terminate the sequence. Very good. Now, we have names for this. Who knows the names? Fancy names. Missense. Nonsense. Yeah, good. Which one is missense or missense? Yeah, good. The way I remember this is, say it however you like, and say missense or missense, as long as you write it in the exam. I remember a changed amino acid and for missense. Make sense? Missense changed amino acid. Okay? That's a missense mutation. What do we call a stop codon mutation? Good. The way I remember that is stop has an O, nonsense also has an O. Okay? Or you can use logic because missense has changed amino acid, the other weird word has to be the stop codon, right? And the last one, you don't have to explain it, right? There hasn't been any effect of that mutation. There has been a mutation, but it has not changed phenotype. The amino acid is the same, the protein will be the same, phenotype will be the same. So do you see how it's silent? Good. Now, this is all written here, but these are my notes on it. Just have a quick look. So as you can see, changed sequence, C went to a G, we changed one amino acid, missense mutation. Nonsense, we've changed a base again, and that's resulted in a stop codon, which has resulted in a premature or short polypeptide chain. The final is your silent, where you still make the exact same amino acid despite the change in your base. Okay? Now, question, which one's the worst? Out of the substitution, out of the 3 substitution mutations, which one is the worst? Very good. Good job, right? It's very important to understand that nonsense is very deleterious. It will probably have the most impairment to the organism's phenotype. What about frame shift and nonsense? The answer is actually it depends. Do you agree? If you have a frame shift, imagine you have 3 codon sequences or 4 codon sequences. Let's say you've got 20,000 and the frame shift mutation happened here. Do you only agree it's only going to affect that next codon sequence? That's it. Whereas if you had a stop codon mutation here, that's going to be an extremely short polypeptide chain. So the answer to that question is it really depends on the location of the frame shift and the stop or a nonsense mutation. Yes?
So would you say all mutations are negative or as in?
Definitely not. You have to think about this, right? There's only one way humanity has generated variation and new traits. That is mutation, right? Everything else you have to understand is a reassortment of traits. Okay? You guys seen Toy Story? You know, Mr. Potato, Mrs. Potato? You learned about crossing over random segregation independent assortment. That's just reassorting traits, like how you can reassort their facial features, you make a new potato, right? That's what crossing over random segregation independent assortment does. The only way you can make a new feature out of the blue is mutation. Does that make sense? So I would say mutation has been integral to human survival as well. It also has the negative effects. Make sense? Good. Any questions at all so far? Anyone? Okay. This section is easier. Chromosomal mutation is quite logical. There's only one that's a little bit weird to understand. What can we do to a chromosome? We have A, B, C, D, E, and F. These are all genes on that chromosome. What can we do to a chromosome to change it? Think logically. Yes, you could delete a certain section, so that's chromosomal deletion. You can insert a section, so that can be chromosomal duplication. Depends on where that section came from. If that section just copied itself into a new section, that is called duplication. If it came from another chromosome, that's called translocation. Trans, across, location. Okay, so we've just identified 3 chromosomal mutations. Good. Now, inversion is a little bit less intuitive, because what inversion is, if you cut a certain section of that chromosome, and you flip this section 180. So where will the F gene be now, relative to this locus? Will it be close or the further gene now? It's going to be here, and the E is going to be here. That is a chromosomal inversion. So when a piece breaks off, there's a 180 flip and returns back. Okay? So as you can see, the little diagrams of it happening. Good. All right. Any questions so far? I have seen it too, but not commonly used. But yeah, if it's in a textbook, you can call it. The issue is dealing with these very simple things, right? We're just adding fancy names to it so we can communicate. So block mutation is a word, and that as well. Good. Right. I'd like you to do one question, which is question 4 here. Question 4. This is one of the most high-yield questions for logics. So out of everything you learned this lesson, this question, I will tell you, is probably the most important question type you should master to answer. It's not hard, but it's an application question, hence why it very commonly comes up. Can the online students, can you all see the question?
Yep.
Yeah. Yes. Oh, there's just one page missing? Oh no, alright. 22 what page is missing? I can get that page sent to you guys. What? It says it right. Oh, the number's different? Alright. Alright, so do this question. It's the same question. Good. Do this question to go, and yeah, I think I imported an older version of your notes, so mine would be wrong. When online students get an answer, raise your hand.
And then we rewrote the rule and MRI, and then we multiplied that with the So that's G, so you start G, and then A, and then later U. So it's that's right? That one? A, yes. And this one, A, right? And then it's E, so then it's U, and then that one, so then it's K jobs. And then same level, based on A, it was. And then yes, this one for the second one. Yes, that's what I mean. Yes.
All right. Lookman, you've got a question or answer?
Answer.
Oh, good. Who's anyone else solved this question? Completed? Let's have a quick video. You need to also focus on time. You need to do these questions in a timely manner. Some of us are a bit slow, which is fine now, but I wish we get faster. One more minute. I guess, Lookman, while you're waiting, you can start doing more of the trial questions or other questions in this booklet.
Yep.
Yes.
Where's the most in terms of
So it could, like I said, it may result in a dysfunction.
Maybe like 2 months ago, I would have been trying to. No, 2 months. How it affects the RNA, right? 2 months. 2 months. 1 month. What's I need you to stop.
All right, let's do this. Question 4. All right, so we've got a DNA sequence. There's a genetic disease which results in the following sequence in the same gene. Now, can anyone point out to me, how would you approach this? You need to have a system, right? When a DNA sequence changes, what's the first thing you want to check? You got the flowchart. What's the flowchart that we just did? Wait, mutation goes to substitution or fraction. So how do we find out if it's one or the other? Count the total number. So very quickly tally up how many bases do we have for the first and the second? Good. So we know it can't be a frame shift. It can be a multiple point mutation or point substitution, but likely not a frame shift if it's 12 and 12. Okay? Definitely not a frame shift. Good. So then what's different? Line up all the letters. G-A-T-A, that's one difference. Any other differences? T-G-C. There you go. So we've straight away identified this is a substitution. And what are the 3 outcomes that can go? Nonsense, missense, or silence. There we go. So that's how you systematically, in 10 seconds, find out what's wrong. Okay? Count the bases and then line it up. Good. What's the next step? So, Raman, how do you interpret this? 5 prime, this 3 prime. How do you go about this?
You would use like the codon chart to figure out what amino acid it codes for, right?
Good. So tell me, this is a codon chart. What does it convert to codons? Does it convert DNA to codons? Does it convert RNA to codons?
It converts the codon, like the 3 base pairs, into an amino acid, right?
Good. But the question is the 3 base pairs. Are we talking about 3 bases from DNA or the 3 bases from RNA?
RNA. So we have to change it to RNA.
Good. So that's the key learning point of this question. Okay? So the key learning point is all of these graphs. You've seen the tables version of this as well, right? We only convert mRNA to codons. We are currently at DNA. So what do we have to do? And when we change it to mRNA, what must we do? That's the word I'm looking for, right? So the key learning point here is all these tables graphs are all mRNA to codons. So you need to always make the jump of converting the DNA to a complementary mRNA. So you tell me out loud, what do we get? Yeah, we'll start with the mutated one. Okay. We can do the correct one as well, but we'll do the mutated one now. C-U-A-U-G. G. Okay, and I guess it's the exact same except for the first, second, third, fourth letter, which will be different. So 1, 2, 3, 4, because U might be different, right? So the original is the exact same, except instead of the U, there will be a A. Yeah, that's the correct sequence. This is the incorrect one, right? Good. So now, the first codon is going to be the exact same, CUA. What is a CUA codon code for? What amino acid? Now, question is, where do we start? Do we start 5' or 3'? 3', 5', 3'. Lookman, where did you start?
I started from 5'.
Good. You can kind of, if you logically deduce it, the 5 is huge. It's the center of the circle, right? Do we understand this though? Which direction does RNA polymerase read the DNA? 3 to 5. Remember, I told you, we read left to right. Enzymes in DNA replication and transcription are going to read in ascending order, so 3' to 5'. When they make the complementary sequence, remember DNA is anti-parallel. If it's reading the sequence 3' to 5', the new complementary RNA strand is 5' to 3', right? That's why we're going to be reading 5' to 3'. Good. So what does CUA code for? CA, which is leucine. Good. Does that make sense? We interpreting it right? Good. What is AUG code for? Okay, interesting. And what does AAG code for? AAG, G's there, is lysine. Very good. So normally we would have coded for leucine and lysine. And what would AUA code for? CUA. Oh, my bad. I've made it very silly. CUA. Let me go back. This is why. What I would recommend doing is circling the 3 codons. Don't do what I did. So that was my mistake. So you're right. So the second codon is UGA for the incorrect sequence. And what does UGA provide us? Stop. Good. So we were correct there. But what would AGA code for? AGA, arginine. And circle the next 3. What would UAC code for? Tyrosine. And what would GGG code for? Glycine. Good. Right? Leucine, arginine, tyrosine, glycine. All right. So what is the effect of this substitution? Nonsense. So yeah, I want you to tell me, where were these 7 marks? It's crazy. Where are you going to get 7 marks from? Where were the 7 marks from? Okay, I get where you're coming from, but the way I'd like you all to structure your answers in a long response is based on the question. What does analyze mean? You should know the HC verbs to a T, because if you know it, it's 3 marks. Alyssa, what does analyze mean? Analyze verb. Quickly, yep. Look, when you're going to be last resort, because I'm quite sure you know the answer. Let's go with Kirtana. What does analyze mean? Quickly.
Cause and effect.
No, cause and effect is explained. Ramit, what does analyze mean?
Isn't it like the relationship between them, and like you kind of you come up with a conclusion at the end?
Good. Getting closer, but it's identify relations and then identify components and draw relations between them. That's HSC word for word what it is, right? So what that means is, what are the key components of each section of this question? What do you all see? Analyze the effect of the mutation on 3 things. What have they asked you? RNA, protein, whole organism. So can't we structure our answer as RNA, protein, and organism. And if it's 7 marks, we can assume it's probably minimum. Just type that. How many marks did you all put to each section? 7 marks. So at least each section is 2 marks, which means how many points per section? Is that not logical? And if a 7-mark question, what would you also want to do? Define key terms. Very good. So add a couple definitions. What are some definitions you'd like to add? He mentioned some really good words. Point mutation and nonsense. Very good, right? So we might want to define point mutation and nonsense. Or even the word mutation might be good to define, because we don't know what the guidelines have. If the guideline has student defines mutation and you don't define it, unfortunately you won't get them up, even if you define what nonsense is specifically. Okay? So this is why you have to be very cautious and kind of cover all your bases. Right? So do you see how we structured this answer? Now you tell me, for RNA, what are the 2 things you want to say? How has the RNA sequence changed? So we can say that there was a point mutation and a substitution. How did that change the RNA sequence? We can write down the RNA sequences, right? So 1 mark would be probably the correct sequence. Everyone agree? Good. Is there anything else we can say for the RNA? Really? It's a point mutation, it's a point substitution of this particular letter. This is a new sequence. That will get you all the marks you need. What about then for the protein? Very good. So we say that we have resulted in a nonsense mutation. We can define it. Stop codon. We can provide the correct codon. So we've already provided the RNA sequence, but we could provide the correct amino acid sequence. So what's the correct versus the mutated amino acid sequence? Remember, this question links to protein. What do we say then? Yeah, an extremely short polypeptide. So we can say another mark for shortened polypeptide. And then what about the final point, which is the whole organism? Let's go up and see if we missed any buzzwords here. Gene, enzyme, very good. Very good. So function is the enzyme will be dysfunctional. And what will that mean? What? If the enzyme is dysfunctional, it's not going to be slower, it's not going to occur, right? So the underlying chemical reaction for an RxN4 reaction will not occur. I'm looking for one word. If you don't say it's one word, you're not getting the mark. No. Oh, you could, yeah, you could say metabolism is altered. What else? I'm looking for one last word to mention.
Phenotype.
Very good. There you go, right? So what I mean, you've got to know the buzzwords for the module. So the phenotype of the organism will be altered. So that's the word I was looking for. Altered phenotype. There you go. Do you see how we broke down the 7 marker? Now, if I wanted to do this in an exam setting, I could probably break this down myself in about minutes, a minute and a half. That's an investment, because you save a ton of time. You won't double up your answers, you won't go on tangents, right? You know exactly what you're going to say. You've thought about it, and you're going to execute it. Very good. Now, I know I've used a lot of, do you see how I use a lot of short words? It's to show you what your marking plan should look like. You should be writing full words and full answers. These are little prompts for me. So I've written shorthand because it saves time. You want to have the shortest, fastest marking plan to get full marks. Does anyone want clarification on what these words mean? No? Makes sense? Yeah. I do it even shorter than this. The way I do it, it should be letters, you would understand it, right? So whatever works for you, but as long as you have prompts, that's what you're going to write for every mark of that long response. I promise you, if you jump in straight to just answering it, it's impossible to get 4 marks. Okay? Good. Does that make sense? Yes.
So when we're defining, can we actually like apply the
Definition in your explanations? Yes, that is an even higher level student. So what I'm saying is just add definition at the top is good, it'll get you the mark, but a really good student will define as they explain. And being able to do that is a skill you build throughout the year. Okay? It's a very good point. All right, let's move on. Here's a literal copy of that question from an actual trial paper. It's from the 2019 Independent. Don't do the question, but I just want you to realize these questions are extremely high yield. They don't care about whether you know about viruses and products and metabolism. Your HC exams came off for about data analysis, okay? Because that's what makes an intelligent student. It's not someone who memorizes a ton, it's someone who can apply knowledge in new ways, which is what the syllabus wants you to do. It's what society wants as well. Good. All right, let's spend the last 5 minutes talking about this point. Now, to talk about this dot point, could I get could you read out the dot point first? 1.3. Can someone quickly read it out? Loud, so the whole class can hear. Okay, to understand this dot point, well, the key point here is what's the difference between somatic and germ-line mutations, right? Now, to understand this dot point, I'll give you guys a small story. There was, I think it was a doctor or a scientist in China who did germ-line editing of children, right? And he cured them of, it was either HIV or it was another incurable disease that he cured them of. Sickle cell disease, one of the two, okay? And he succeeded. He cured a disease that humanity's never been able to cure before. But he also did something that was illegal, which was germ-line editing. Now, does anyone know what germ-line editing is? So when we talk about genome editing, right, you can either edit DNA in a somatic cell, so that can be a skin cell on your body, so it's a diploid body cell. It's already divided, it's differentiated, it's a mature cell. A germ-line cell is found in the gonads. It's your spermatozoa and your ova, right? Now, that's germ-line. Germ meaning refers to your haploid cells. Now, if you edit a diploid cell, you stuff it up, will that be inherited to offspring? Think about this. Will the skin cell on this part of my arm be inherited by my children, or will it be the germ cells that the individuals carry? It's a germ cell, because it's a spermatozoa and the ova that will combine to form a new organism. Make sense? So somatic editing doesn't have many implications. If you stuff it up, the organism is stuffed up, they don't pass it on. But if you germ-line edit, if you edit the spermatozoa of a male or the ova of a female, you stuff that up, they will pass it on to their offspring. And remember, that ova of spermatozoa is going to form every cell, right, of that zygote multicellular organism. And then that organism will pass it on, and that organism will pass it on. The implication is germ-line mutations are indefinitely carried on, whereas somatic mutations, they die with the organism. Does that make sense? Now, germ-line mutations are what you've already studied. Do you remember autosomal recessive, autosomal dominant, X-linked recessive and dominant? Those are all due to gene or chromosomal mutations that are germ-line. They're passed on. Does that make sense? So I'm just linking it up for you. Okay, good. So in this dot point here, somatic mutations, they're mutations in diploid cells, right? So it's any somatic or body cell of an organism. It's not passed on. Give you an example, skin cancer. Someone gets melanoma on their skin. Does that mean if they have a child, the child will be born with melanoma? No. So you intuitively understand this, right? The skin cell does not form the new organism. It's the ova, the spermatozoa of the parent. Okay? So that's what the syllabus wants you to understand. Germ-line mutations, they are passed on. An example of this is Down syndrome, right? In Down syndrome, let's use a little diagram to understand this. Maybe we'll use, I'll drop myself. Yeah.
So you guys said skin cancer can be possible. Would something like ovarian cancer be possible?
Yes, but it's not the cancer, but it's a mutation which is passed on. So we'll come back to this very shortly. We have 2 minutes, so I'll quickly talk about this. So this is an artistic drawing of a person, right? If you have a somatic mutation, that is in their body cells, okay? Their specific germ cells, whether it be spermatozoa in the ova, are unaffected. And so when they form a new organism, the new organism is healthy. Whereas if you have a germ-line mutation, such as Down syndrome, an individual with Down syndrome has a trisomy 21, or 3 copies of chromosome 21 in every single cell. The skin cell, the hair cell, the sperm cells, everything has mutated copies of chromosome 21. So that means the spermatozoa mutated. And so when it combines with an ova, it is going to form another organism that is entirely carrying that mutation as well. Does that make sense? This is your example of a germ-line mutation. This is your example of a somatic mutation. Okay? Yes?
Can individuals have they possibly gene and possibly recessive? If they breed with the mother?
Yes, it can. It definitely can. So it could be that the mutation is recessive and it gets masked by a healthy copy from the other end, right? But the potential is it can indefinitely be passed on. You're completely right. Go back to inheritance patterns. It doesn't have to be masked by another dominant allele. But see, it depends. When we're talking about dominant and recessive traits, we're talking about gene mutations. Do you all agree? Down syndrome is a whole extra chromosome. Things like that can't be masked. Okay? So that makes it a little bit more complicated, but that's the gist. Now, I'm going to teach you about germline and somatic again next lesson. So when we start, we'll run through this again because we really rushed it. And we'll do a couple of exam questions so you know what they'll ask you in the exam setting. But otherwise, we're done for today.